GEMM core does is we make genetically modified mice so that investigators can have an in vivo model to further their research to understand the environmental impact and further the NIEHS mission.
We develop the strategy, we make the mice for you. We don't stop there. We can help you to develop the most efficient strategy so that you can get your data for publishing the paper.
I really enjoy about my role that we are producing novel mice that have not been produced in the field before. We're making the first genetic mutation for a lot of the projects that we work on. That is extremely fulfilling because we see that months of hard work and labor went into this, and we finally have the end product of a gene-edited mouse that we can then pass on to the investigator to use.
It's invaluable to us. Our research would be completely impossible without something like the GEMM core that can make genetically modified mice custom to order, essentially. Our research is completely dependent on that.
By doing it in-house, we can do it significantly faster. We work very closely with the investigators. Sometimes they don't know necessarily exactly what they want coming in, and that's where we can help them figure out exactly what they need in a mouse.
The fact that we're in-house and we're readily available allows us to really make sure that any kind of help that's unexpectedly needed once we've generated a mouse line, that we're here to help. And genetically, we know it best because we're the ones that made it.
The person that we really interacted with is Artyom, who was really excellent at explaining what the issues were, what the possible solutions were to the problem that we faced. It became clear pretty fast that we were really fortunate to be able to have someone with Artyom's level of expertise to figure out what needed to be done.
It's huge factor for investigators, those who are coming to the institute, not having each lab have their own settings, because it is impossible to have each lab with these very expensive instruments and on the top of that the expertise. Many of them don't have this expertise.
The timeline to making a new genetically modified mouse model is long. In the days before CRISPR and other targetable nucleases, we would typically be talking about 2 years from concept until there was the first founder line. Now with CRISPR, that timeline has shrunk to months, but it hasn't changed anything about the life cycle of the mouse. You're still limited at the gestational and reproductive life cycle of a mouse. Now on average, I'd say we're down to about a year from concept to a stable mouse line. So most of the mouse lines that we're publishing now, we made 5 or 6 years ago, and the investigators have had for 4 years.
We work for the investigator on a day-to-day basis, not just making the mice, but guiding them how to use them. Any mouse-related project can take years before they publish the paper. So it's very expensive to maintain those mouse lines, but we help them to have the most efficient strategy to reach their end goal.
It's awesome being able to come in and get a little tidbit of what each investigator here at the Institute is doing. And when we make these genetic modifications, I get to see in a year or two down the road what they actually did with these mice after they expanded the colony. And that is fulfilling to see that something that we made probably 3 years ago is actually being put towards scientific studies.
Unlike other cores here at NIEHS, it does take a considerable commitment from the end users group, because it will be years to really fully study whatever mutation we make for them.
Having a team that's going to be working with you for the long term can really be essential. When you have the cores in-house and they're citizens of the Institute who expect to be here for the long term, it's a genuine collaboration where both parties really care about the science and about advancing your understanding of it and making for the best possible outcome.
If each investigator spent the time and money to get proficient at the microinjection skills, then there would be a lot of time spent of their lab doing that. So that's why it's beneficial for us to specialize in these skills, that it saves them time and they can just use us as a core facility.
They can definitely take the advantage of using outside companies, but that is very expensive and their support is one term. If it works, works, if it doesn't, doesn't. And they hand you the mice. There is no help in using those mice and how to set up the colonies, how to breed them, because those are extra things.
Our happiest day is when we let the investigator know that your mutation has gone germline. We have successfully generated your mouse line.
I do enjoy the work. I enjoy the variety the work brings. It's not one thing on a daily basis. It's something a little new every day. And that's interesting to me.
So I'm very lucky in the sense that I honestly can say that I don't have a job. I get paid to play with puzzles. And every different allele and every different mouse project is essentially, it's a brand new puzzle that no one has ever done. And it's just a completely novel aspect.
The GEMM core has been, one of the most important cores for our research. They've been unfailingly helpful and have been able to do everything we wanted them to do, even when it's complex or somewhat out of the ordinary.